Hi, I'm Jack Cush, Executive Editor of RheumNow.com. It's the 3rd of February 2017, and this is the RheumNow Week in Review. A number of interesting items on this week's agenda. A recent report in A&R from Jeff Curtis and colleagues looked at the risk of infections associated with the receipt of a biologic, in this case, Remicade. They looked at a large Medicare population, looked at claims data, and looked at how close these patients received their Remicade infusions to their actual surgery, and whether or not it resulted in a higher risk of hospitalized infections or within 30 days, or post-operative joint infections within 12 months. And it looks like it didn't matter whether the patient had received the biologic, in this case, again, infliximab, within 4 weeks of the surgery or later time points, 4 to 8 weeks, after 8 weeks, and even after 16 weeks. So on one hand, it looks like this data says that all the fuss about infections and the timing of surgery may not be all that important, but there are some caveats here that you need to be careful. Number one, this is claims data, and there's no reasons given as to why patients had their infliximab held longer in some places and shorter in other places, and what were the factors that were involved in that. Secondly, the drug that we're looking at here is infliximab, which is a long-acting TNF inhibitor. In fact, if you want to wash out a TNF inhibitor, it takes at least 4, if not 5 half-lives, and the half-life of Remicade is 210 hours. So it may not matter, given the half-life of the drug, for these results. In general, they saw very low event rates that were, I think it was 6 to 7 per 100 patient years. It didn't, and it really didn't change very much. So while this is good data, it's not foolproof, and I still think we need more good studies like this to know about the timing of surgery. My rule is suspend the biologic for 1 dosing interval before you do the surgery, and that means 1 week for etanercept, 2 weeks for the Q2-week drugs, et cetera, et cetera. Another report looked at the utility of chemokines and its correlation with disease activity in those with untreated early rheumatoid arthritis. And they showed that a battery of chemokine, CCXL10, had the highest correlations with pretty much all the clinical parameters we would use for activity, all the DAS measurements, CDAI, swollen joint count, SED rate, CRP. All these were the best correlated with CCXCL10. So CXCL10. Again, I bring this up. I brought up calprotectin in the past as a biomarker, a potential biomarker, and I still think the search for an ideal biomarker is in process and needs yet to be defined. Something better than SED rate and CRP and good clinical assessment. Good news on the biologic front. Health Canada was the first to actually approve the new IL-6 targeted drug, sarilumab. Clinically or commercially, this will be known as Kevzara. This comes from Regeneron and Sanofi. It's been submitted to the FDA and the EMA for approval. Its approval process was a little bit sidetracked in the United States because of a manufacturing inspection issue. So instead of being first quarter, it may very well be second quarter when sarilumab or Kevzara is approved in the United States. But it is available now in Health Canada at a dose of 200 milligrams every other week. We wrote about this a few weeks ago in an article called The IL-6 Wars. I guess is the beginning of the IL-6 wars. Analysis of gout and how it's treated was looked at by a group of Swedish investigators looking at a Western Sweden population and correlating serum uric acid levels with the onset of gout. And they basically showed that compared to levels of less than 6, levels of greater than 6 were associated with a levels greater than 10 serum uric acid were associated with a 70-fold higher risk of developing gout. And for those who had gout, serum uric acid levels greater than 9 had a 5-fold higher risk of developing recurrence and flares. Interestingly, even when the uric acid levels were less than 6, the incidence rate on flares was about 12%. So that's interesting news in the world of gout. A study of 1,300 patients with psoriatic arthritis showed that psoriatic arthritis patients may have a 1.43 odds ratio or hazard ratio of developing diabetes. So a 43% higher risk of developing diabetes if you have psoriatic arthritis. This feeds into one of the big concerns with psoriasis and psoriatic arthritis, and that is the development of comorbidity. This risk seems to be linked to inflammatory parameters such as joint counts and ESR levels. Another national nurses' health study, a large population-based study over time, shows that postmenopausal women have a 2-fold higher risk of developing not just RA, but seronegative RA, which I found really interesting. Again, it's hard to tell what some of this the nurses' health study data tells us because it doesn't have any explanation. It's hard to link to other factors. They also showed basically that early menopause was also associated with a higher risk of rheumatoid arthritis, a 2.4-fold higher risk. And again, that was mostly seronegative disease. A nice and interesting study looked at what goes on with hip and knee replacements. In the United States, in the last year or so, it's about 1 million Americans will have hip and knee replacement, but the complication rates are about 1 in 12. And unfortunately, what this article went on to look at was, is there a predictive parameter, a profile? And unfortunately, there are no predictive parameters or tools to identify the high-risk patient, one who will have postoperative complications. A study from Daphne Gladman's group looked at actually, it was Daphne Gladman's group that looked at the incidence of diabetes. There's another group here that actually looked at the risk of fracture in psoriasis and psoriatic arthritis and showed a 2-fold increased risk of fracture, especially vertebral fractures. What happens when you switch in from Remicade to a biosimilar Remicade, in this case CTP13 or Inflectra? And a study recently published showed that when they looked at interchangeability, meaning switching from the originator to the biosimilar, they showed no differences with regard to ACR 20, 50, and 70. This would be the Planetra study. And also no differences as far as immunogenicity and anti-drug antibodies. These are a few of the steps that are required now with the interchangeability provision that's been provided by the FDA. Previously, we have right now 4 biosimilars on the market. 3 of them are TNF inhibitors. None of these were given an interchangeability indication because there was no guidance document that just came out. But this is one of the studies that does look at interchangeability and is important to define interchangeability. A meta-analysis of lupus patients and adherence was really quite sobering and showed that at least half, and on general the average was around half, but ranged from 43 to 75% of lupus patients were non-adherent with their DMARDs and medicines meant to treat their lupus. Maybe even more sobering was the fact that 1/3 of them were off of such therapies after 5 years. Lupus is hard enough to treat without having to deal with non-compliance and non-adherence. This is the big, ugly, hidden monster in clinical care these days. Dealing with the issues of non-adherence and non-compliance, we need better tools to look at this. A study of patients with psoriatic arthritis who became pregnant showed that while their skin disease does well, their joints may very well worsen. So again, when you start to look at pregnancy and rheumatic disease patients and using objective parameters and not just what you last remembered, patients often don't go into remission. In this particular study, up to 40% of patients had some degree of flare during the follow-up period. A review of the long-term safety of adalimumab or Humira was again published with Gerd Burmester as the lead author. Gerd has written now, this is his 3rd review of the adalimumab database, and is always very informative if you want to get a real picture of what biologics can do, and especially adalimumab and other TNF inhibitors. What I found interesting in that report was the pregnancy data associated with adalimumab. Again, over 15,000 patients reported they had, I think, over 70 pregnancies that they had good data on, and there are only 3 documented congenital anomalies with less than a 4% malformation rate. And when they compared that to other RA patients and non-RA healthy controls, this rate was lower. I mean, those other rates were around 4 and 5%. So that's good, encouraging data for those patients who do need to take a TNF inhibitor during pregnancy or leading up to pregnancy. I had a question about the occurrence of lupus, and I did a little bit of a literature search and found a cohort study of 103 patients that supposedly had lupus polyarthritis. Of course, that would be patients who meet criteria for RA and lupus and are seropositive for both. In their review of 103 patients, 10% of their SLE patients had lupus polyarthritis, and of those patients, they were less likely to have renal disease and obviously more likely to have erosions and higher joint counts, higher SED rates, and CRPs. An article that was published in the literature looked at gout and how it's been managed, and it was really sort of shocking. Actually, this is the study that comes from Western Sweden, and what they showed in the first year of being diagnosed, 32% of their gout patients actually received urate lowering therapy, which is in general somewhat of a low number, but not unreasonable. But what was unreasonable was that at 2 years, 75% of them were not taking urate lowering therapy, meaning that there was very low persistence with such therapy, and that's such is the case with gout and males who are stricken with gout. These rates were higher in males. These rates were, as far as those who received urate lowering therapy, higher in those with diabetes, cardiovascular disease, and renal disease. Again, a real problem with long-term persistence and the management of gout. A review of non-steroidal use in spondyloarthritis was a bit telltale. This is actually a small study, but a prospective study of like 50 or 60 consecutive patients that looked at the use of non-steroidals. What they did show was that non-steroidals were highly effective in the clinical parameters you would look at for spondyloarthritis, but actually had no effect on SED rate, CRP, and MRI measures of activity and inflammation. After 4 months, there was still a healthy percentage. Almost half the patients still had a BASDI of greater than 4, and up to 30% of patients had to reduce the dose of nonsteroidals, largely because of intolerance to nonsteroidals, obviously well known. In the end, only 13% of patients stayed on nonsteroidals over a long period of time and stayed on them at maximal doses. So again, I think this sort of dispels the potential myth that nonsteroidals should be a mainstay of therapy in ankylosing spondylitis or axial spondyloarthritis, because it seems like they're not well tolerated, and certainly not well tolerated at the doses that are needed. A review by Janet Pope and Roy Fleischman and a number of different leaders in the field looked at an analysis of all the studies that compared combination TNF inhibitors and methotrexate to the triple DMARD therapy. So this would include studies like those done by the RAIN Network and Jim O'Dell, the TIER study and the RACKET study, and also the SweetFOT study. But there was many studies, in fact, over 30 studies that were actually looked at. And they looked at patients who had established disease who received either one of those two regimens. And they looked at DMARD naive or early disease, methotrexate naive patients who received either of those two methods. And what they found was that really there's very little difference between the two. If there was a difference, when looking at an ACR 70 as the primary endpoint at 6 months, there was a slight edge for those with established disease, and that they were those on triple therapy were less likely to achieve an ACR 70 at 6 months compared to those on the combination biologic regimen. But when you looked at the same analysis in early disease, it didn't seem to pan out to be different. Safety was really not much difference, although there were maybe a few less infections in those on the triple DMARD therapy. So while this network analysis, meta-analysis was supposed to, I think, maybe make a case for biologics. I think it was underwhelming, if you ask me, suggesting that there may be a role for triple DMARD therapy. And lastly, Cassie Calabrese wrote a nice review of an article that looked at the utility of the pneumococcal or pneumococcal vaccine, PPSV23, in an RA patient cohort. And after a prolonged period of follow-up and compared to those who didn't get vaccinated, there was no difference, with about a 3 to 4% association with infections in either group. It could very well be that the way this was given was not in accordance with the current guidelines, where you should get the Prevnar 13 followed 8 weeks later by PPSV 23. And in general, research on this area with the pneumococcal vaccine, it's very hard to prove the benefits of it in almost any population. So a bit sobering. I don't think it's going to dissuade me from using the pneumococcal vaccine in the future. I still think it's important to do it according to the ACIP guidelines, which say that you start out with Prevnar 13 and then do the PPSV 23 8 weeks later, and then repeat that second one, the Pneumovax PPSV 23, 5 years later. That's it for this week with the RheumNow Week in Review. Tune in next week for more good news on RheumNow.com.